Welcome Genetic Teacher channel. Today, we are talking about bacterial cells counting, which divided to three types. 1. Direct bacterial cells counting, to count viable and ideal bacterial cells. 2. Viable bacterial cells counting, to measure effects of bacterial growth. And 3. Bacterial cell mass measurements, to measure effects of bacterial growth as well. Firstly, direct bacterial cells counting, divided into three methods: light microscope method, flow cytometer by spectrophotometer method, and filter membrane method, by calculating growth colonies on membranes, as mentioned in the next 3 slides. While viable bacterial cells counting, including 3 methods as well, by spread and pour plate methods, filter membranes method as well, and serial dilution method, to distinguish between living and dead bacterial cells, as mentioned in the next 3 slides. On the other side, bacterial cell mass measurements, do by weight bacterial colonies in solid media, or bacterial cells in broth, in form of pellets, for analysis metabolic activities, and turbidity, as mentioned in next slide. Additionally, this slide demonstrates bacterial colonies counting, by bacterial counters. And this slide display bacterial colonies counting by camera scan. Thanks for attention. Please like and share and subscribe Genetic Teacher videos and channel.